Alright, this mini lecture we're going to start with this graph, and this graph is used a lot in Chapter 8 and to introduce the principles that are described in metabolism in Chapter 9 and later in our course, Chapter 10 in photosynthesis. It goes like this. We've got a chemical reaction. In this case, the substrate or the reactant is this molecule AB and CD, like that, and there's going to be a chemical reaction, and we're going to end up with these molecules interacting with each other, leading to products AC and BD. And so it's general, and here's just the chemistry of it. There would be a reverse reaction like that. And so here we have the overall chemical reaction, and that's represented on the x-axis, and it's referred to as the progress of the reaction. It's a little bit like time. The x-axis is a little bit like time, where we start at the beginning and we see the reaction go forward to the products. The reason it's not exactly like time is because of this reversibility. So the reactants can flow to the products can flow to the reactants, like that. And you can see that we drop, so we're going to flow downhill. And this is where the analogy with water flowing downhill is imperfect, because when water flows downhill, there's never any water, like a little bit of water that's flowing uphill. That happens in chemical reaction, and it's called the principle of microscopic reversibility. As a result, reactions typically don't go to completion. So we can start out with 100% of this material, and we rarely end up with 100% of products and 0% of reactants. And it's because of the reversibility. So it's common that we might end up with, say, 95% at the end and 5% here. And at the end, by at the end I mean at equilibrium, and people can refer to this as final equilibrium. So the reaction starts out with 100%, boom, and over time product starts to accumulate, first a little bit, more and more and more, and then we can let the reaction go till final equilibrium and see how much of each is represented. And how much of each is represented at the end is based on how much this drops. If this drops further, there'll be a higher percentage of this guy at the end, because if it drops really far, this microscopic reversibility becomes more and more rare, like that. So this change here, and change in math is referred to by this symbol delta, and this is important to mention in studying the chemistry of life. And the y-axis is referred to as free energy, and free energy has the units of calories per mole or joules per mole. So this is the axis where we're going to measure how many calories are in the sugar or in the fats we eat, like that. And so here would be fats up here. They have a higher potential energy, a higher free energy, the sugar here. It's actually hard to measure the total free energy in a molecule, but it's relatively easy to measure the change that occurs during the reaction. And this free energy is named after a scientist who worked on it a lot, whose name was Gibbs. So this is often referred to as Gibbs free energy, and this value is often referred to as delta G for the change in Gibbs free energy. And delta G determines the final equilibrium. Okay, now here's the next thing about this graph, and that is that this does not determine the rate of a reaction. The rate of the reaction is how fast this thing happens. Like that. When you're burning wood in a fire, right, the rate of reaction is very high. The rate of the reaction is actually determined by this. So this is interesting because, I've been drawing it, and I was just drawing like that. Boom, there's a drop, the water flows downhill. This represents sort of the fact that the water's in a bucket, that it can't flow downhill, that it's got to get a bump up over this to go flowing downhill. And this delta G here There's a delta G here, right? And that's called the activation energy. Woo. Activation energy determines the rate of the reaction. So we got two measurements here we can do on the y-axis, the delta G for the reaction itself, and that's going to determine the final equilibrium. So that could be lots of energy released from the fats, you know, lots of energy released in the fire, like that. That's that. Then this delta G, the activation energy, is how fast the reaction rate would go. If the delta G for the activation energy is very big, the rate of reaction will be very slow because it's very unlikely to get this up over that hump and down like that. If it's low, the reaction is going to happen pretty fast. And what happens in the wood of a fire is, you know, the way wood burns is other wood burns and heats up the wood. So it's kind of like a chain reaction where the burning wood heats the other wood and it catches on fire. The way that happens is that, remember how we said water holds heat in the cup of water by vibrating. And so as the wood is heated up, it's holding this energy at a higher level. So the starting reactants of the chemical reaction of burning wood are actually at this higher level, and it becomes more and more likely that they'll jump over that and boom, down to the other side. So that's burning wood in a fire. You actually raise the potential energy of the reactants by heating them up, like that. Then something happens very different in our bodies, because our bodies don't burn calories by heating up the material, right? Our body temperature doesn't get burning hot. Instead, here's what happens in our bodies. The activation energy is lowered. So the activation of this chemical reaction is lowered, increasing the rate of the reaction. And so that's how life works in this. And the way it works is that enzymes function as catalysts to increase the rate of the reaction by lowering the activation energy. So that's a key concept there, is that you can burn glucose in a fire. This is how it's done. You can burn glucose in your body. This is how it's done. So two different things. Notice that this doesn't change. The delta G of the reaction doesn't change. The final equilibrium doesn't change. The energy released doesn't change. So it doesn't matter how you burn the calories. How you burn the wood, the same amount of calories are released. So when you burn wood in a fire, it releases this amount of calories. When you burn the same material in our same glucose, we can't digest cellulose, but we can digest glucose. Burning that same amount of glucose in our bodies, the same amount of energy is released through these two different strategies. It's like, you know, this is a pathway through the chemical reaction, and the pathway doesn't matter. You get to the same end point, like that. You start at the same place, end at the same place, and the path in between doesn't matter. Okay, so we'll talk about this graph a lot, and we'll use this. We'll use this shorthand a lot. All right, so here's an example of how does the activation energy get lowered. And we already looked at this reaction where we take the substrate sucrose table sugar, and we're just going to cut that by a chemical reaction of hydrolysis. And the way our body does that is that it typically it uses an enzyme, a catalyst, and this purple molecule is typically a protein. So now imagine a truck in Boston, and the truck has on its side a pocket about the size of human body, like that, and the pocket has a specific shape in which the substrate will bind, and that pocket is called the active site. So again, we have a big truck protein, and it's big in order to hold the amino acids in this active site in this very specific position, so that this active site will specifically bind this substrate. And so enzymes in our body are specific. So consequently, we have thousands of different enzymes because each enzyme has a specific substrate binding to its active site through non-covalent bonds, and then the enzyme catalyzes the chemical reaction. And how it does that is it provides an alternative chemistry. So you remember we talked about how water can be destabilized by bringing another water molecule near it. And that's what's happening in the active site, is that this sucrose is normally very stable because it has a big activation energy. So it's not just going to spontaneously break apart like that. And that's a kind of key to life, right? Sucrose is a plant sugar. The plant made that to store energy, but not store energy forever. And so here's the key thing. This hump is the key thing. It's a way to store the energy so that the trees don't spontaneously catch on fire in the woods, but when it's time we want to release the energy, we can. We can because this hump can be gotten over, and the way cells get over it is this. They lower the activation energy when they're ready. So when they're ready, they use this enzyme to bind to the substrate and start to digest it, start to break it down and release energy like that. So you can store energy and release it. And that's a big, big key concept in life. And once again, we have these proteins playing a central role. And it's because proteins can take on so many different shapes. We've already seen several shapes, and shape determines function. And we've got a function here. And that is that this active site will have a chemistry where there'll be non-covalent interactions. You can see a little bit of the structure of the sugar. There's going to be some hydrophobic, some hydrophilic regions, and the substrate binding site, the active site, matches the chemistry of the substrate. So it's very specific. Typically, when the substrate binds, it's not shown here. When the substrate binds, the protein typically changes shape, and people refer to sort of a lock and key mechanism, where as the key fits in, the lock is changed and opens the door like that. Once the enzyme has catalyzed the chemical reaction, here are the products, and they are released, and the enzyme can act again. And so enzymes are catalysts that are not used up in the reaction. They're recycled. They can be used again and again and again. And typically, the turnover number, that's the rate of the chemical reaction, is 100 more or less per second. So 100 of these chemical reactions can be catalyzed by one protein molecule a second. Boom, 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 like that. So enzymes are very active catalysts, and we'll see what we have next. Here's just a zoom in of sort of a more indication of the shape of the active site and how when the substrate binds, the active site and the overall protein change conformation. The key thing is the protein's bringing alternative chemistry into the substrate, encouraging it to undergo a chemical reaction. And we're going to stop here, and we'll start with this figure on the next mini lecture and talk about the chemical catalysis of proteins in our bodies using this graph.